to one and all. We once again welcome you all to the today's session of virtual workshop on computer-aided drug design. Now the session will take over by Professor Mukesh Doble. Sir,
Yeah, okay. Good morning, everyone. Today we will talk about the target-based design. So, past 2 days we looked at the ligand-based design. That's the drug which goes and binds to the target. So, we looked at quantitative structure-activity relationship. That is how we develop a mathematical relation between the structural features or structural descriptors with the activity. And then we looked at pharmacophore-based approach, which is identifying the features, special features, pharmacophore features of molecules by superimposing them, like whether they have common hydrogen bond donors or acceptors or hydrophobic groups or aromatic groups or cations, anions. So that's called pharmacophore based modeling. So the left hand side is all related to the ligand based design, okay, or lead based design or drug based design. That's the small molecule. On the right hand side we have the target based design. That means I should have some idea about the target protein or enzyme wherever the drug is going to bind to. That means I should have the three-dimensional structure of the target. So generally, how do you get three-dimensional structure? It's through X-ray crystallography. The protein is crystallized, and then they use a X-ray diffractometer to get the coordinates of the various atoms. Okay, so it's a time-consuming process. The sample has to be very pure, and if it's a protein taken from a medical situation, then the amount may be very less. So it's a long process. Whereas the sequence of the protein could be obtained much easier. You purify the protein maybe by using 2D gel electrophoresis, and then you will get the sequence by using either ESI, MSMS, that means electron spray, ionization, mass spec. So one mass spec will fragment generally the amide bonds get fragmented. That is C double bond ONH2. That's called the NH is the amide bond. So that will get fragmented. So you get fragments of small peptides. So from the two fragments, you start building up the sequence of the protein. So the sequence is much easier to get, whereas the 3D structure, structure is much time consuming and longer. And on top of that, you should have some idea about the active side, and that also is much longer. Actually, it takes a very long time. So we can't just blindly assume any pocket as the active site. So that also is a time-consuming process. So sequences of millions of proteins are stored in a database that's called the UniProt database. UniProt, so a lot of sequences are stored actually, millions of sequences. Okay, this is the database. So if I have to compare my if I have a new protein which I have taken from my research, I got a sequence, I can compare the sequence of that new protein with proteins existing in the UniProt database. Then I need to use this actually, understand? So we use this, it's got millions of sequences available, and as I said, it's much easier to get sequences. All you will need a 2D gel electrophoresis to separate out the proteins, and then you will require a mass spec, ESI type mass spec. So it's not very simple, although it's much easier than getting the three-dimensional structure of a protein. Okay, so you have many features in this. Okay, so like BLAST is a program which compares the protein sequences actually. So you can see this. So it's quite it gives you a lot of help. So it's very simple. Okay, so we just give the sequence. We write down the sequence. Suppose I have a protein which I have sequenced, or a small peptide which I have sequenced. So I put it in this box here, and then I will say search, run blast. It will try to search. Okay. So look here. Enter either a protein or nucleotide sequence of a uniprot identifier. And then it will start searching, and then try to say what is the sequence similarity with the existing. For example, they themselves are giving actually P00750. So we can put that, for example, P00750. So that's an existing protein sequence. But if you have your own protein sequence or a peptide sequence, you can put it in the box. Okay, the amino acids. Then you say just run blast, and then okay, so it's running. So it takes a little bit time to run that. So you can see it takes a little bit time. It will compare with various sequences of proteins in this UniProt database, and then tell what is the sequence similarity. So it will give you in terms of percentage, 30%, 40%, 50%, 80%, and so on actually. So in this case, it may give very high because I'm putting whatever the number is there. So each protein that's put in the database has a standard number, UniProt number. So that is the sequence of this, and then it is doing the job. As you can see here, it's going around. There is another. So this is related to sequence database. Similarly, we also have the 3D structures of proteins. Okay, that's a PDB, protein data bank. Okay, this is the protein data bank. So we have the UniProt database, which gives you the sequence of proteins. Generally, you will have millions of sequences, but when you go to 3D structure, when you go to 3D structure, you might not find so many. You may find maybe few lakhs only, because getting the three-dimensional structure of a protein is much more time-consuming than that. So here we can search for the 3D structures of protein by putting in some number. So for example, I put COX-2. Okay, COX-2 is an enzyme which is in the inflammatory pathway. Okay, and it is involved in arthritis, pain, and so on actually. Okay, so it's given different COX-2s. Why is it? Because many researchers would have crystallized these COX-2 with the different ligands. Okay, so whenever they are, and then they get a 3D structure, and then they put that 3D structure. Number one. Number two, it could be COX-2, that is cyclooxygenase 2, from different sources. It could be human, it could be animal, it could be rats, and so on actually. So that's why you will find so many cyclooxygenase 2 enzymes actually. Okay. And again, you can see it's bound to different ligands. Okay. So this is a human, as you can see, the human COX-2. So you will find murine, you will find so many different things. Animal sources, basically from different sources. So this is from bovine heart. Okay, so like that, again, this is from bovine heart. So you'll find different COX-2s from different sources, and of course there will be some differences in the sequences. So depending upon the type of research you are doing, you can take the particular 3D structure of the protein for your research. Okay. So different sources and with different ligands bound to it, sometimes in closed form, that means there is no ligand bound, or sometimes in open form. So you can see lots and lots and lots of COX-2s. Okay, so it's not, then again there is another page next and all that. So this is bovine heart that you can see oxidase. So different types of COX-2s are stored in this particular so the beauty of this database is it also gives you how it was, who deposited, is there any corresponding publication, so you can go back to that publication, read the paper, and then get an idea about the research through which this particular sample was taken and through which the 3D structure of this um, protein was determined. Okay, so this is the protein. So if I want to carry out any docking with the small molecule or a ligand or a lead molecule or a drug, we can download this, and we can once we know the active site where the interaction is, we can do the docking. Okay, so we can download the corresponding, in this particular case, COX2, depending upon the type of study we are working on. Actually. So if you are looking at human COX2s, or you are looking at some other species COX2, so you download the corresponding COX2. Okay, so it gives you lots and lots of these COX2s. Okay, so the UniProt which we ran, it's given, as you can see, it compares with different proteins and comes up with sequence. So it will give you lots of comparison, going right up to 90, then 82, 85, almost hundreds. So it gives you from Homo sapiens, tissue type, plasma. So it gives you the sequence comparison with the different okay, protein sequences, as you can see here. Okay, so the program which does this job is called BLAST. Okay, so that you might have studied in your bioinformatics course. Okay, so depending upon which is the highest sequence identity, we may take that sequence, and if the 3D structure of that sequence protein is available, we build the 3D structure of our protein using that, and that is called the template. So template is the protein 3D structure whose sequence is similar to ours. So similar means you will not get 100% similarity, even 35, 40, 50% similarity is okay. So that is called the template. Okay, then we can build and then we can run. Or if the protein 3D structure is available in PDB, then well and good. We can download the PDB structure and then we can do our docking. Okay, so the docking comes later, but initially, if the 3D structure is not available, then we look at the protein which has got sequence similarity similar to our protein through the when you run it in UniProt. We take that, and if the 3D structure of that protein is available, we use that as the template, and then we model our protein. So we get the structure for our protein, that's called homology model. Once we get the 3D structure for our protein based on some template, then we can dock your ligand to that. Okay, so for docking, there are many softwares, commercial softwares there, free softwares are there. So free softwares like Autodoc is there, Vina Autodoc Vina is there, Swiss Doc is there. I'm going to show you Swiss Doc, much easier. Commercial, there are many commercial softwares are there, so that's not the focus of our thing. For homology modeling, generally things like BLAST, which does the homology modeling. But I will show you Swiss model, which does both BLAST and then finally get that sequence of the protein of interest. Actually. Okay, so we do the docking. So we have a set of molecules. We dock all of them to our protein. Either structure of the protein is available or we have modeled our protein. So we dock them and then see which ones dock very well. So take those ligands, neglect those ligands which don't dock well. And then, of course, we finally have to do our filter using drug likeness properties, Lipinski's rule, blood-brain barrier, bioavailability, toxicity, ADME, and so on. Actually, so that is the workflow for target-based design, which we do. So generally, these UniProt and PDB take a very important role. Actually, so if you know the 3D structure of your target, then life is easy. So it may be available in our PDB database. Okay, so 3D structure of our target is known. Excuse me. So we just download the 3D structure. Then I have my ligands, set of ligands. Say I have identified some 100 ligands using either pharmacophore modeling or QSAR. So I can dock each one of them into it and then see which ligands dock well, which don't dock well. So when you say well, maybe we can compare it with some control. So if there is already a drug reported for that, we can keep that as our standard and see how that drug docks, and then we can see how our molecules dock with respect to that. So either through docking score or through binding energy. So you get generally negative values for binding energy. More negative means better is the interaction between the ligand and the protein. So we can keep that if there is a standard drug which is reported, we dock that first. So that's very important. So whenever we dock our other molecules, we see whether these new molecules have better binding score or more negative binding energy or less. So we can neglect those which don't seem to dock well. So we can take those which seem to dock very well with respect to the standard. So you should also always have some standard to compare with. Okay, once we select those molecules, then we apply our drug likeness property and then remove those which don't satisfy. Okay, so if the 3D structure is known, then all we have to do is download from PDB, and it's like finding a key that fits the lock. So you have many keys. So some keys open very well, some keys open little, some keys don't open at all. So throw them. So it is like that. Whereas if you do not have the lock itself, okay, that means I don't have the 3D structure of my protein. I have 3D structures of protein with similar similarity percentage. Then I take the 3D structure of those, that's called the template protein. Then I build the 3D structure of my protein, that's called modeled protein. Okay, and then I start making keys which will open the lock. So it is like first building the lock and then finding the key. So in the first case, it is like just finding the key that fits the lock because lock is already there. In the second case, because we don't have the protein 3D structure, so we make the lock from locks of similar type, and then we start making keys to open the lock. So that's called, first part is the homology modeling, second part is the docking part. So first part is homology modeling, second part is the docking. So homology modeling becomes very important. In many cases, this will happen because if we are looking at human protein, if you are designing drugs for disease related to human, you might not find the corresponding proteins for human, but you may find the corresponding proteins in other species, rat, mouse, murine, dog, so many things. Okay, so we may have to do the homology modeling. So I may have the sequence for the human protein, but I might not have the 3D structure. I may have the sequence and 3D structure for some species. So I will do homology modeling, that is sequence comparison, and then I will use that as the template and build my target protein. Okay, so in many cases in our problems, we will be doing homology modeling. Because for human diseases, many of the proteins are still not crystallized. Why is that? Because the amount of protein may be isolated from a human is very, it could be very small. Whereas in animals, we can trigger or we can develop those type of animal models, and we can have enough amount of protein to be recovered, purified, then crystallized. But for human, that's a big problem. So in many problems, we may end up doing the homology modeling. Okay, so it is like building the lock, finding the key. The concept of homology modeling is that during evolution, the structure is more stable and it doesn't change as much as sequence. That means even if the sequences are quite different, the 3D structure remains similar. That's very interesting. This has happened in evolution. So even if 30-40% sequence similarity, sequence means the two-dimensional, the 20 amino acids, ATGC, all those amino acids. Okay, so even if there is only 30-35% sequence similarity, still the proteins seem to attain the same 3D structure. That's very, very interesting. So in fact, it's very good for our bioinformatics. We can take proteins which has about only 30-40% sequence similarity with our protein sequence, and we take the 3D structure, the template, and start building. Okay, so that's very useful because of this interesting human evolution. So this was identified by Lesk and then by Schneider. Okay, both the groups identified that. So distinctly related sequences still fold in similar structures. Okay, there could be some differences maybe in some other place, but generally they adopt, especially in the loop region or the external region, but still they adopt. Okay, this is an interesting picture. So number of aligned residues, percentage of identical residues. So if I align say 150 residues, okay, and only 30% or 40% of them are matching, okay, we can say that it's good for homology modeling. Okay, so if you are aligning or comparing 150 residues between protein A and protein B, okay, if you are getting say more than 30% identical residues, then we can say the structures of those proteins will be conserved. Okay. Only if you are aligning very low number of residues, that means if protein A sequence and protein B, we are comparing only 50 residues, or then we need to have more than 40, 45, 50%. But if you are comparing more than 100 or 150 residues, even 30% identical residues, percentage 30%, then we can say we can use the 3D structure of the other protein to build the 3D structure of our protein of interest. Okay. So if you are comparing almost 150, 200 residues, then around 30-35% is good enough. We can say that both the proteins will have similar structure. So, and that is what we will do actually. So up to 35%, below 30%, it's not safe. Above 35%, it's good enough. Okay, so there could be situations where we may get 35-40 only. So we will not get 80-90 at all, especially when we are looking at real life problems. Okay. So you are comparing the sequence a protein A, which has got 150 amino acids long. Okay, then we have another one B, which has got 300 amino acids long. So comparing the 150 of that, and 50% are identical. Okay, so it's good. We can take the structure of B, which is called the template, and we can build the structure of A, which is called our model protein. Okay, so that is the important concept in homology modeling. So in this graph, anything above that, that means it's safe to consider the structure of that template protein to build our model protein. Okay, so proteins are conserved. So that's a very interesting thing that has happened in evolution. So many faraway proteins with a little bit more than 35% sequence similarity, you'll find a similarity in structures. Okay, so what we do is we have to recognize the template. That means we have to align them. So UniProt Blast is the software which does that. Then we develop our model, and then we can do some corrections. We can do molecular dynamics based studies. We can do molecular mechanics based studies. All those things can be done. That's called backbone generation, loop modeling. So we get your model. Once we get the model protein, we do the model validation. That means we see whether the protein is good. What is the root mean square deviation with respect to the template protein? We also draw the Ramachandran plot. The softwares nowadays do all these things, but you need to know this. You must have studied Ramachandran plot long time back in proteins. The phi psi angles should be within the allowed region. So many, and then there is a what if check. So there are many checks to see whether the protein which you have modeled from the template is good. Okay, so there is no clash between the amino acids and so on. Okay, so once you have checked all these things, that protein, we call it model protein, and use it for your studies. So you can dock various ligands which you have shortlisted maybe from your ligand based study, either QSAR or pharmacophore, and then start working on it. Okay, so UniProt is the very important database which gives you a lot of sequences, okay, which I showed you here. So we put in the program which runs is called BLAST. So we put our protein sequence or peptide sequence or whatever we have isolated in our research. We have got the sequence using a ESI MSMS or MSMSMS. Okay, so we put in that and it compares with the existing proteins, and then you can see the picture here. It compares with the existing proteins, and then it gives you a score, high the score. Okay, and then also it gives you a percentage. In real life, you will not get 100%. Here, this is an example of existing protein sequence itself. That's why you got 100%. Okay, otherwise, so as you can see, different numbers are there. It's not that always you have to take that protein which is highly 100%. It depends on your problem. So if you are, say, doing studies with certain rats or murine, so you would like to take the sequence, the similarity with the proteins isolated from similar species. Okay, so it is not that it always have to be the highest number which you have to take, but it should match the similar species depending upon which one. So we can take that. Okay, so it gives you a lot of protein comparison, as you can see here. So numbers goes to 91, 92, so many pages. It's given many proteins. Okay, so it's identified about 248 protein sequences which has good sequence comparison. So you'll get starting from 100 right down. So that's what UniProt does, and this is what the program which does is BLAST. You must have studied. I think biotech students studied in your bioinformatics course. So we can take that, okay, that particular thing, and then if the 3D structure is available, we can use that as a template. So it gives a lot of information about the proteins which has been compared with, actually, as you can see here, the left hand side. Okay, so that's UniProt. Then the PDB I talked about, which is very important, which gives you the 3D structures. This PDB database, it's called Protein Data Bank. Sorry, Protein Data Bank. Again, this gives you information about the literature related to how the protein was isolated from which species. All publications, all the informations are given. So you can read them and see whether it matches with the research which you are planning to do, whether it aligns with that. So you must always keep that in mind. The research which you are doing, either on a particular species or disease related, so you should match with that particular PDB protein or UniProt protein. Okay, this model is combines which combines both the UniProt part, UniProt part, as well as the homology modeling, getting the target protein. So say start modeling. So here we can give the protein sequence. Okay, then we'll say search for templates. Okay, then once it is identified, we'll build for model. Okay, we'll see that. So sysprot does that. Sys model. So let's look at this particular thing. Prostaglandin synthase 2. This is an enzyme in the prostaglandin pathway for inflammation. Okay, it's so we are looking at this particular thing. Okay, protein sequence. This is from mouse. So I can input that. Okay, so it's got the so it is already existing, that's why it's able to get the sequence. But if it is a new protein sequence which is from your own research, we need to give the protein sequence. Please remember that. So because I have taken from the existing PGH, then we can say search. So we can say search for template. So this is the protein sequence. I hope you can all see that. Then we can say search for template. Okay, so it will take a little bit time because it has to go through millions in the UniProt. So it will take, so it's going on and on and on and on. So it will try to identify, and again, and just it's run through BLAST. So it will give you proteins sequence similarity starting from highest percentage, or E value will be the lowest, and then it will give you in that order. So it's working. So it will take some time. So it's currently running. It takes a little bit time. So once we get the sequence similarity, so then we build our protein with the protein which has highest sequence similarity. So that will call be template, and our protein which we are 3D structure which we are building is called the model protein. Okay, that is the activity just follow. It will take a little time. Okay, so it's done. Not bad. Fast. So it's given. It gives you a lot of information, as you can see here. So it gives you the various template protein structures, okay, and then gives you the title, and then it will give you the sequence identity here in this column. You can see, okay, and how those proteins are crystallized. Sometimes they use X-ray crystallography method, sometimes they use NMR method, okay, and then what is the percentage accuracy, okay, smaller the percent and the smaller the accuracy, that means more accurate system will be, okay. So a lot of information, as you can see, it's a dimer. That means you have two proteins homodimer. It is called. You can see here. So you can see this is 100% identity, 100, and because it's existing, you'll get. But if you are running your own problem, you might not. Like I said, you may get much lower. If you have your own protein sequence, which you have obtained from your experimental isolation of the protein, and then MSMS. So this is a prostaglandin synthase 2. This is called a cyclooxygenase. So the protein is crystallized. Okay, and information is done. Okay, so this is much better. So the protein is crystallized. Then this is also a prostaglandin H2. This is a murine. As you can see, the third one is murine. The first one, there is a palmitic acid, okay, bound to the protein. So somebody has put in palmitic acid, and then the protein is crystallized. So generally, if you do a ligand bound, the active site of the protein gets conserved. I mean, remains. Otherwise, when you crystallize and there is nothing in the active site may collapse. That is why you should look at whether active site some ligand is there or not, so that we can be sure that the active site has not collapsed. Then we can remove the ligand and put in our various lead molecules. So this is again a murine based. This is a human based. You can see here. So you see different sources. Here aspirin bound to the human cyclooxygenase. So if you're looking at human cyclooxygenase, this is a good protein to select. This has got only 88% similarity, but then this is better because I work quite a lot on inflammation. Aspirin is one of the non-selective drugs which are used for inflammation. So this is very nice, which is related to our research. So depending upon the research which you are going to do, you select the particular template which matches. So don't just go by the percentage similarity, but select the protein which matches your research. So I may take this. I can see here. I will not take the 100% similarity. I am taking actually 88% similarity. Please remember that. The PDB-ID of the protein is given. It says the crystal structure with aspirin bound to it. So X-ray is collected from X-ray. So as you can see here, so I have selected this protein as the template, and then I can start building my 3D structure of my protein. That's my target. We call it target. Okay, now we can say build model. So I select only one. Sometimes we can select 2, 3 like that also. So it will take all the 2, 3 and then try to build your protein. Okay, by taking all the. So in this case, I have not taken the topmost 100% identity. I have taken much 88% identity, and then I say build a model. Okay, so it will build model. This particular protein is taken as the template. This is a cyclooxygenase 2 prostaglandin synthase 2 with aspirin bound to it. That is taken as the template, and my protein is built whose sequence matches with this particular protein at 88%, and then it builds my target protein. It's doing that. So you can also have 2, 3 templates together and then try to build your protein also. That's also allowed. Or you can take just one protein and try to build your protein. That okay, it's done that. It's doing fast today. Okay, so it has done that. as you can see here, it's done that. Um, So a lot of structural assessment. Like I said, we need to know whether the modeled protein is okay. So this is the alignment, as you can see here in this picture down, and alignment between both the protein, the one which I have given sequence, and the protein which we have selected as our template. And then it gives you a lot of details about the structural assessment, how good the assessment is. This is the protein here, which is shown here. We'll look at that little bit later. It will give a lot of information like quality checks, comparison, as you can see here. Sequence identity is 88%. Like I said, we are not looking at 100% because I'm more interested in human inflammation, so I have taken that particular. Okay, structure assessment that we can see already. It's giving here. Quality, just call it global quality assessment. Structural assessment. Okay, so I talked about the Ramachandran plot that is called the sci-fi angle. As you know, Professor Ramachandran worked in Madras University and Indian Institute of Science, and he came up with the angles which the amino acids take. That is the phi and the psi. Phi is the second dimensional angle, and psi is more of the third dimensional angle. So um, he found out that you cannot have any angle. There are only certain allowed region into which these angles have to fall, because if it is outside the allowed region, there could be some contact between one amino acid and the subsequent amino acid. So he defined some regions which is called the allowed region, and so you have to see whether all the five psi angles of the protein which you have modeled falls into it. So you can see that most of it falls. There are some um, um, not so fantastic region, but still this is also okay. Little bit is okay. This is best, and this is not so best. So there could be some clashes of this. So we can okay. So you can take that. We can later on do something called force field molecular mechanics optimization. So they will go away. So we take this protein, we can do molecular dynamics or molecular mechanics, force field optimization. So the protein will definitely adjust itself so that the clashes between the amino acids will go away. Okay, that can happen, but you can see majority are falling inside the allowed region. So it's a good model which we have done. So if the majority falls outside, then you are in trouble. But majority falls inside. So like I said, we can do geometry optimization as it is called. Then we can even do molecular dynamics. So when we do all those, the amino acids get realigned so that the clashes are completely eliminated. Especially some of these you can see here. Okay, some of them here, and these are slightly away outside. This is not so bad region, but still this is the best. Okay, so it gives you the structural assessment. This is called the Ramachandran plot. Okay, so and then it gives you a lot of this is called the clash score. That means the amino acids, successive amino acids which are clashing are given. That is here. These outside ones, whereas those inside are not clashing. Okay, so it gives you a clash score. So it gives you Ramachandran outliers. So not bad. It's up to second decimal, it is 0%. So that's also very good. There are some bad bonds here, bad angles also. There are some bad angles. It gives you those places. But when we do a molecular dynamics of this protein, hopefully they'll all go away. Okay, then you use that particular protein for your docking studies later. So you need to look at this. Quality check is very important. Okay, then it also gives you the RMSD values. Okay, so there are not everything is within this line. As you can see here, everything is within that line, so it's not bad. There is not much deviations in this protein. Okay, so everything is within this line. You can see here for all, it's we're doing for the entire chain. You can see chain A. Okay, so for the entire chain, it is giving you these values. So that's the residue quality. So we look at Ramachandran plot. We used to also have that. So this Q mean is an indication of this is the residue number. Okay, so this gives you comparison of non-residual PDB. So it gives you the see there are one or two only which is outside, and most of them are within the region. Okay, so you can see this one or two regions which are slightly outside. Then this gives you their okay. So local similarity with the target and template protein. So only in some places it's not similar. Okay, if the deviation is large, that means some places, those places residues are not similar. But in this region, the residues between the target and the template is, you can see, it's similar. Here. You can see here, the deviation between the target and the template is not similar. So these have deviations. Okay, so this is a very useful quality check which gives you the structural assessment of the protein. So now you have a protein, it's reasonably good. There are a few okay, by side where there is clashes. So hopefully when we do molecular mechanics optimization, they will all go away. Okay, and the E value is good, and the Q mean value also all falls within this, and the percentage of bad angles are quite low, as you can see. And there are very little Ramachandran outliers. So 96% of the amino acids fall into the favored region of Ramachandran plot. Only 4% are outside, that is these points are outside the favored region. But hopefully they will go away when I do a molecular dynamics. Okay, so you need to do a good quality check of the protein which you have generated. That is the modeled protein which you have generated from the template. So this is this set of activities which we need to do. Don't forget that. So just don't blindly go into homology modeling, take a protein, then start talking. We need to have a good understanding of the quality of our protein which we have created. Okay, so this protein I can download, and I will say that this is my modeled protein which I will use it for all my studies. Okay, let's go back to our presentation. Okay, so this is the approach by which we do the we take the sequence of the protein. We can put it in the Swiss model, as I showed you. Yeah, we can put it in the Swiss model. Yeah, Swiss model. So it does a sequence comparison. As you saw, the program which does that is called BLAST, and then we take the template which has a higher percentage of sequence similarity, or which is more relevant to our research. In this case, I showed you I'm more interested in human inflammation, so I took a template which has only 88% sequence similarity. Then it builds the software automatically. So its Swiss model is very good to do that. It can do a sequence comparison, and then it can do a building. So it builds your protein, and then once it has built, you look at the quality of the protein which is built, how good it is. That's very important, using Ramachandran plot, Cumin plot, and then bad angles. Okay, and then you say that your protein which you have built is good. Then of course later on we will do some geometry optimization, as it is called, or molecular dynamics, to relax the protein and get a very good structure. So that structure we use it. Now this particular protein has got aspirin in the active site, which is good. So we can take that protein. We know where aspirin is inside. So that will be our active site. So that's very good. So that's going to be our active site. Uh, so we will know that will be our active site, and then we can remove the aspirin, and then we can put our drugs of interest, which we are working on, various ligands which we are working on. Okay, so this is the modeled protein. Yeah, as you can see here, aspirin is here. There could be other ligands also. When they crystallize, they get many things into. Aspirin is here. Acetyl salicylic acid. You can see the aspirin here. As you know, the structure of a chemical name for aspirin is acetyl salicylic acid. Okay, so you can see acetyl salicylic acid somewhere here. Okay, so of course there are many things inside, because when you crystallize, there could be a lot of impurities also getting stuck to that. So advantages of having a ligand with the protein crystallized is, we will know where is the active site, so we can remove that ligand, and we can start putting our ligands. If we do not have any already existing ligand, then we will not exactly know where our active site is. Okay, so that problem is going to be there actually. This is some other ligand. So many impurities are also stuck with the protein, so that is why all these are coming inside. And sometimes if there is a cofactor, then that also will come inside. Okay. Okay, so this model does a good job of getting a homology model of your protein of interest. Okay, that is, we saw the sequence of activity. So once we get the protein of our interest, we go to docking. There are many free softwares like AutoDock. Vina is there. AutoDock. Again, SwissDock is also there. I will show you SwissDock because it's much, it's easier, it's faster. Whereas AutoDock like programs, we need to spend a lot of time to understand the workshop. So with a limited time, we will just look at SwissDock. So I'm going to show you SwissDock. 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 Yeah, SwissDock is this program. So here we give the as you can see here, we give the target, that is the protein, and then here we give the ligand, that's your drug or lead molecule, and then here we give here we give job name, and then here we give our email ID. So the data, the results will come to us. Okay, so let's run in some examples. So I'm running aspirin with cyclooxygenase 2 enzyme. Again, this is an inflammatory. So 1PXX is the protein in the protein data bank. Okay, so that is the cyclooxygenase 2, and then I'm going to run aspirin with using SwissDoc. So here I give the target. So I'll say 1PXX, 1PXX, search. So it has found 1PXX. So if you look at the PDB, so it shows, yeah, this is 1PXX. This is the crystal structure of diclofenac. Diclofenac is an anti-inflammatory given for inflammation bound to cyclooxygenase and the active site. So this is good. The advantage is if you remove the diclofenac, you will know exactly where the active site is. So we can put in our ligand. So in this, okay, so this is yeah, this is the one with the diclofenac inside. So I have selected this for my study. Okay, so SwissDoc, yeah. So we say this. It's got 4 chains. So let's take a chain. Okay. Of course, we should have some background knowledge about where the active site is in which chain. But here I've just taken, then select for docking. Okay, so it's selected, it gives you a tick, that means everything is okay. Now I'm going to put aspirin, ASPR, that is enough, I think. Search. Yeah, so you can see acetyl salicylic acid. This is the acetyl group, this is salicylic acid. So I select this ligand. So if I have many ligands, you can select many. Dock one selected ligand. So it does some check and then say, it's done. Okay, good. So I'll give some title. I say aspirin doc to cox2. Then I give my email ID. Then I say start talking. So it will take time. Start talking. Yeah, it will take some time. I've done this docking. So I will show you. Okay, and internal error. Okay, oh yeah. Good. That's an internal error. So let us redo the whole thing. Luckily, I have done this docking. Yes, 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 yes. Start talking. Experts will do it again. Okay. Yeah. Aspirin search. Aspirin search. Dock on selected molecule. Aspirin also done. Yeah. Then start docking. Okay, so it's gone. It's gone to my. It will go to my email. So it takes some little bit time to get the results. So as you can see here. Okay, so it will take some time for the results to come. So it will take some time. I already done. So yeah, so it will take some time to come. Started. I have already done the results, so we can see um, aspirin doc2 cox2 results are here anyway. So to view these to view the, so this is how the results will come. To view the protein, we need to have viewer called UCSF Chimera. This is also a free software. It's called the UCSF Chimera. So we need that software to view the protein. And so it gives you the cyclooxygenase 2, and it's given, as you can see here. It puts the ligand in different places, in different locations on the protein, and then it calculates the delta G. So you can see many numbers are there. So we can have hundreds of places placed also. It will take longer time, of course. Okay, and we can view. Okay, so it has placed your aspirin, as you can see here. It is placed here, and it calculated some energy. It comes to minus 7.43 kilocals. Then these are all one group. So so it has placed like this, also in a different place, as you can see here, and that gives you minus 7. So it gives you different kilocals, some very good. So you also have minus 5.8. So here, so it is, as you can see here, it is placed here, and energy value comes out to be minus 5.8. So we can run many like this. Minus 5.5, minus 7. So minus 7 point something looks the best. Okay, minus 7.43 looks the best. So this position, aspirin looks the best. Other positions gives you different numbers here, as you can see. So we can look at all these locations. So as you can see, it's moved to some other place. So it keeps the ligand in different positions, and then calculates energy. You can see that. So it's moved to some other place, like that it does. We have minus 4.8 also here. So that's not very good. Yeah, here as you can see here, so that energy is not very good. So somewhere here, I think it looks the best. Okay, so that's how. So as you can see, different large number of positions. We get different energy values. Okay, so we can do it for different ligands and get the energy values. Okay, so here, as you can see here, gives you 4.89. So this is a quite reasonably simple software. So we can place your ligand in different places, and then see how the energies look like. Okay, similarly, I also did another. Okay, we saw aspirin with COX-2. So tripnavir. Okay, this is an antiviral drug. this goes and binds to the 3CL protease of SARS. Okay, so 3CL protease of SARS, there is a crystal structure available, so we can run that also. So I can give the protein 3CL and search. Yes, so it is found 1 PDB. So we can look at that protein. So this protein is 3CL protease, which is involved in the replication, actually inside of the RNA of SARS. Coronavirus. So you can see this. Okay, so this protein is a coronavirus main protease. There are another protease called PL protease, also papain-like. This is a 3CL protease. So they are this is a target for anti-SARS drug. So we will look at that as our protein of interest. So it's got many loops or chains. So we'll take one chain. We later on have to look at other chains also. Okay, so it's taking that, and then we will take Navir. Okay, so we will take this is an antiviral drug. It's reported for 3CL protease SARS also. So I am taking that as my ligand. So we'll get the zinc ID. It's there. I think it's there in the zinc ID also. Let's see. Okay, it is there in the zinc ID also. View the zinc entry. Tripnavir is there. Remdesivir is not there in the zinc. So this is tripnavir. It's there. Even if you look at drugs DB, bang. Okay, so there is a sulfonamide containing hydroxy. So it's a non-peptidic protease inhibitor that targets the HIV protease. It's been given actually. Again, ritonavir is also given for HIV protease. Okay, so it's antiretroviral treatment of HIVs. Tripnavir was also given for the current pandemic also in some cases. So we can give that and see how it docks to the SARS protease, which is 1P9U. Okay, so I picked up that, and then I similar thing. I say 3CLP tripinavir. Then you have to give your email ID. Then you say start docking. It'll take a few hours to do the docking. So tripinavir, it's a sulfur. So it's a sulfur containing compound. There's a sulfur group is there. Then SO2. This is okay, as you can see here. This is sulfur containing. So tripinavir is a non-peptide HIV protease inhibitor. Okay, so we will go to again. Okay, so it has gone, and it will take a few hours. I had done it some time back, so we will have a look at that result. Yeah, so this result. So here, docking prediction area will be here. Okay. So this is the protein. Okay, protease 3CL protease of SARS, and this is your ligand tryptophan. You can see here ligand tryptophan. It's a bigger molecule. Sulfur is there, sulfur dioxide is there, so it gives you a lot of energy values, -9 point something, -8 point something. So it puts the -9.97 as the top here, and then there is -7 point something, like that. It keeps giving different numbers. 8.77 something is there. So this -7 point something here comes out to be better. Okay, so it's giving you different numbers. Okay, even 6.93 is there. That's a low number here. So these places are all not so good. Somewhere on the top loop, it looks good. So bottom places are not very good. So if you look at the top places, so this place appears to be good, where the number comes out to be -9.98. Okay, so this is only one loop, so we can do for the other loops also, and then see how the binding energy. So basically, we look at binding energy. Some softwares also give binding scores. So binding energy means lowest number negative value. Binding scores means highest number positive value. So okay, so that's how the concept of docking goes about. So you start with the homology modeling, because you might not have the 3D structure. So we first compare the sequence of our protein, which we have isolated from some research, with the existing protein, and then we see which one has highest sequence similarity, and then we will take the 3D structure of that protein, which is called the template, and then we build our protein, which is called the target. So Swiss model building is a very good software which does both this job together. So homology modeling. Okay, and then whatever modeled protein, we have to see how good the quality of that modeled protein. So the whole concept of homology comes because it's been found that if you are comparing 100 to 200 residues, and if you get more than 35% similarity between these two sequences, then we can assume that the structure is conserved. Okay, that is how the evolution has gone. So even if you get 35% sequence similarity, if you are comparing more than 100, 150 sequences, then we can say the structure is conserved. So we can take a template and we build our model. So we use the Swiss model. The UniProt database contains the 2D sequence of millions of proteins on peptides and small macromolecules. PDB databases contain the 3D structures of maybe lakhs of protein. Then I showed you examples of how Swiss model is built. And then docking. There are many docking softwares. I showed you Swiss dock. So in Swiss dock, we give the ligand, we give our protein. So the PDB ID of the protein we give. In this exam, I showed you two examples: aspirin with cyclooxygenase 2, Tripinavir, which is an antiviral drug against the 3CL protease of SARS-CoV-2. That's PDBID is there. So I try to talk. So we will look at the energies, and based on the energy, lowest and negative energy, we decide on the best binding partner. You need this graphics software called UCSF Chimera if you want to view the structures from the Swiss doc. Okay, if you want to view the protein with the ligand, we need that, or we need something called JSMOL. One of those two viewers we need, JSMOL or this UCSF Chimera. Okay, one of those two things. As it is mentioned here, either you need a UCSF or we need JSMOL, one of those two things. Then only we can view this proteins also. Okay, so this is how the concept of docking goes. So once we have many ligands, we can keep docking, look at the energies. Lowest energy means good is the docking, and we always have to compare it with some drug so that we will know what is the docking score of that drug or binding energy of that drug, so that we can say our molecules are better binding or not so better binding. Okay, so then there is another software that's called target prediction. That means if I given a molecule, what are the possible targets it may go and bind to? Okay, that's called a Swiss target prediction. This is useful if I am looking at side effect profiling. So if the molecule goes and binds to too many targets, that means called Swiss target prediction. So here I paste a smiles notation here, and it will give us the various possible targets. So again I will do where I think I have we need the smiles notation of where. Yeah, smiles notation tripnavir is here, so I can copy the smiles notation of tripnavir and then go to Swiss target prediction. Yeah, Swiss target prediction. So this can do for Homo sapiens and some species of rats and mouse. So we can. So this is tripnavir here structure. Then we can say predict target. So it gives you possible targets. Okay, so it gives you percentages. So as you can see, cathepsin, proteases. So cathepsin, we have proteases here. We have pepsin protease. So 100 probability, very high probability of this, and other probabilities are low. And then of course this gives you a summary of all the possible. So it gives proteases, okay, 33%, then some electrochemical transporters, large percentage of this. Okay, so this target prediction gives you an idea about possible targets which the molecule will have. So this is also a very useful thing to do. Let's do for another molecule target prediction. Remdesivir is also one of the antiviral drugs that's being looked at for this particular COVID infection. The smiles notation for remdesivir was not there in the zinc database. So, yeah, so I had to use, yeah, I could not get the Smiles mutation zinc database. Maybe it's under some other name. So what I did was, uh, yeah, I got it from drug bank. You can see Smiles mutation somewhere is, yeah, Smiles mutation for remdesivir is there. I could not get it from zinc because maybe it is under some other name. So I got it from drug bank. So I put in, yeah, this is remdesivir. So I said predict target. The remdesivir is one drug which is being currently looked at as possible for possible treatment of COVID-19. Yeah, so remdesivir, it's done. So you can see again protein coupled receptors and then kinases and so on. So, so adenosine receptor, its probability, but still it's not very high. Okay. This is all related to human targets. There is one, sorry, there is another resource, okay, that's called, it's meant only for COVID. It's come up recently. Um, Given a molecule, it can try to predict possible targets by binding them to various targets, and then it will arrange them in that order. Okay, so this is a recent one which has come. Just for looking at targets for the and COVID. Okay, so they keep on adding new targets. Okay, which came up recently. So what we can do is target prediction. We can say, okay, so here we can give a molecule and it will dock to various targets. Okay, it will dock to various targets and then it will arrange them in order of highest binding energy. So it's got many COVID targets, and then it may try to bind to them. So we can upload the ligands in SDF file or MOL2 format, but it will take much longer time to do. It will take several hours. It's a very slow server. So it tries to, based on the binding to various targets, that's as of now loaded. They will arrange it in the order of highest binding energy to the lowest binding energy. It takes long. Okay, so here we can the ligand here, okay, and then choose, and then we can run. So it will go through all these SARS-CoV target and then come up based on the highest score. Okay, that's called D3 docking. There are other. It also identifies the target based on the similarity of the structure of the ligand which you give against something else. Also, the ligands which are stored in. Okay, so this is a recent addition which gives you target prediction focused on SARS-CoV-2. Okay, so the results will come out later. We can look at the results. Um, okay, so there is a remdesivir file is there. Yeah, right. I think a long time back. Okay, so if suppose we run remdesivir, it gives you different types of possible targets, and it also tells whether it is human or SARS. It arranges them based on the score. Okay, so based on the highest binding score, and so on, so on, so on. So if there are any SARS-CoV targets, so we have compiled that. I'll show you the result. So look at remdesivir based on target prediction. So minus 11.09 is the highest score. This is a human target, and this is a SARS-CoV target. You see, minus 10.53. This is the PDB7BV2 is the PDB ID of that particular protein, RNA-dependent RNA polymerase. Okay, the most probable could be human. Next probable could be SARS, and then it again arranges them, as you can see down, based on the binding energy, minus 11, minus 10, minus 9, and so on. Actually, again the remdesivir. If there is another approach, this is the 3D target. Other one is based on the similarity. So they look at the similarity of remdesivir with molecules which are known to bind to different targets, and based on the similarity, it will again try to predict. Okay, that's based on the ligand similarity approach. That's called. Again, we can see remdesivir binding to RNA polymerase, and so on. So this particular this particular website is very useful if you are interested looking at targets of SARS-CoV-2. Recently created a few months back. They keep adding new targets, and most of the targets protein may be created through homology modeling, because I don't think many of the targets have proper crystal structures. Okay, so most of them may be homology modeling. As time goes, maybe they will come up with the 3D structures next 1 year, 2 years, 3 years. So this also gives you some idea about possible targets into which your ligand may go and bind to. So we can look at Swiss target, which predominantly gives human and certain murine and other species. We can dock your you can take your ligand and see which are the most probable targets. So we saw that, and similarly this particular site also gives related to SARS-CoV. So we can use all of them for possible target predictions. Okay, so as you can see, it gives you possible as human target, possible as COVID target, and so on. Okay, so for your Swiss doc, as I mentioned, you need that program called Chimera, UCS of Chimera or JSMOL to view the proteins. Otherwise, you will not be able to view the results of Swiss doc. You have to remember that. So if you want to view the results of Swiss doc, we need okay, so if you want to view the protein with the ligand, we need to have either JSMOL or UCS of Chimera. Okay, so with that, we complete this computer-aided drug design workshop, which we have been looking for the past 4 days. Okay, so we saw a lot of things in the past 4 days related to several websites, several web servers, okay, all freely available software for getting properties of molecules, experimental properties which are already stored, theoretical properties which can be calculated, because structural descriptors, features, ADME properties, drug likeness property are very, very important to prove or disprove whether a molecule candidate could be a drug or no. So that's very, very important. That's why I spent on the first day. Then second day we looked at ligand-based drug design. There are two approaches mainly: the pharmacophore-based approach and the QSAR-based approach. In the second day we looked at pharmacophore. We identified common features or common pharmacophores for given set of molecules. The pharmacophores are hydrophobic groups, the aromatic groups, the hydrogen bond donors, acceptors, okay, and then cations and anions. So we used PharmaGist. So we used PyMol to view all the pharmacophores developed. Then we used PharmaGist to screen new molecules from zinc database which may have the same pharmacophore. Then later on, of course, we have to use this is ADME to see whether the new molecules which you have shortlisted have the drug likeness and ADME properties. Okay. Then yesterday we looked at QSAR. What is QSAR? Quantitative structure activity relationship, and how do you build models, almost like regression models, between the structural descriptors or features with the activity, one descriptor model, two descriptor model, linear models, non-linear models. So you can do many of them with Excels, and you can all. I showed you another called build a QSAR, which can do many of those jobs. Then there is a lot of statistics we need to keep in mind to prove the models which you have developed has good fitting as well as predictive capability. And then today we looked at target-based design. Target means your protein or enzyme which your drug or lead molecule is going to bind and stop its action. So protein 3D structure has to be available. If not, we look at a protein of similar based on similarity, percentage similarity, identity. We take that protein structure, we call that the template, and then we build our 3D structure of our protein, we call it the model protein. So the Swiss model building is a good, which does both. The first part, sequence similarity comparison, which is done by BLAST, and then the model building. Once you build the model, you look at how good the model which you have built using Ramachandran plot, using the all the factors, okay, RMSDs and so on. And then you may have to do some relaxation using molecular dynamics or molecular mechanics so that the protein is has no clashes at all. The example I showed, 96% is within Ramachandran region. There's only 4% which is outside. So once we have the built protein, we do the docking. There are many softwares, free softwares like Autodoc or Bina, and then SwissDoc. I showed you SwissDoc, and then we saw the ligand placed in different places, what the energy is. So we can run this type of study and look at the best energy. That means best means most negative energy. So I showed you some examples on that also. Then came target prediction. This is useful because you want to know whether the molecule you design has going to bind to different places, including human targets, which can lead to side effects. So this target prediction is there for human or for murine and other animals. And I also showed you on SARS COVID, which is recently coming up, target prediction. So we can give our query ligand and then it will arrange them based on the most negative binding energy. So we will know which is the most probable target, second, third, and so on. And the most important thing ultimately is all the filters of various drug likeness rules, blood-brain barrier rules, then ADME rules, bioavailability, polar surface area. So we always have to check that whatever molecule we design either experimentally or whatever molecules we design on the computer screen, we have to check the possible drug likeness property. That's very, very important. I spent on the first day because 96% of candidates fail because of that. Okay, for some reason or the other, because of all these reasons. Okay, so that's very, very important. Simultaneously, we need to do. So this is there are many other things which can do, which I did not talk about. One important thing is the pharmacokinetics and pharmacodynamics. So if a drug is given orally, it slowly gets absorbed. So the concentration may go up a little bit in the blood, okay, and then slowly it will fall down as it gets degraded and then excreted through urine and other routes. So you can model this also. It's called pharmacokinetics and pharmacodynamics modeling. I did not talk about it. It's much more advanced. But we can, generally these things can be seen when you do animal studies. So what they do is they give the drug orally to the animal and keep monitoring how the drug concentration changes with time in the animal. So they get the peak increasing and decreasing. Okay, so they do it in animal, then they can do it on a human also. So this is called the pharmacokinetic graphs. We can model this also. That depends on how good the absorption is and how much fast the degradation and excretion takes place in the body. So it may come out very fast or it may remain longer. So this graph is also very, very important. So there are some models which one can look at to do that. Okay, so we did not talk about it. So I have a 8 weeks NPTEL. I hope you have heard about NPTEL, which conducted by IITs. These are free videos. So I have a 8 weeks NPTEL course on computer-aided drug design, which talks about many more softwares, a lot of theory behind all these softwares. I have not spent time on theory behind. If I start spending, then that could take a very long time. So that NPTEL course on computer-aided drug design, which is coming in the July semester for 8 weeks. I think it's postponed a little bit. So it talks about a lot of theory behind. Uh, then it talks about optimization. It talks about quantum mechanics. It talks about this pharmacokinetics, pharmacodynamics. So all these. That's why it takes much longer time. So I have not covered, any of them the theory behind it. Okay. So with that, I will conclude. Thank you very much for spending. Whoever has spent last 4 days. I hope you have picked up some of these concepts. I hope you will be able to keep seeing the videos so that you can download all the softwares I talked about, the Marvin, the PyMol, the ESF Chimera, okay, all these important ones. And you can practice some of these examples which I have done also, and you'll get some idea about how to use all these. And all these softwares are good enough for research purposes, undergraduate teaching, postgraduate teaching, because we have published papers using these software. So it's not that you need a commercial software to do things, but this is good enough for most of our drug discovery type of research, most of it. And there could be some specialized situations which this might not cover. Okay, thank you. Thank you.
Thank you so much, sir.
Can you hear me, guys?
Yeah, sure, go ahead.
So I want to say that thank you for very much for your lecture, sir. It is a great privilege to listen to you for over the NPTEL courses and the classes and all these workshops, webinars, and everything. It is a great privilege for the other students and everyone. And you are on behalf of St. Peter's College. It is we are honored to host you. Thank you very much, sir. And I would like to thank.
So if there are any questions anybody has also on this docking, I'll be pleased to answer. A few minutes.
Hello, sir.
Yeah.
Yeah, this is Clayton Fernando. I have a question in my docking, sir.
Okay.
I have a target that is amostin for leishmania disease. I had chosen this because it has a essential role in its survival. But I could not find the target in PDB, so I predicted the structure using Swiss model. But in that, I find it out almost 3/4 of the protein are loops only.
Oh, okay.
So, is it possible to target that protein?
Oh, loops. There are no active site, is it? There are no pockets.
Yeah, only one alpha helix is present. So, how can we proceed that target for the next work? Next to
Yes.
take that.
So, two things. One is there any literature already available where somebody has done protein modeling for this situation?
No. They just
Absolutely nothing.
reported this is essential for its survival only.
Nobody has done any protein modeling, is it?
No, sir.
There is no literature at all.
Yeah.
So you need to don't take the loop region for your docking. You have to take the alpha helix only for the docking actually.
Okay.
One is you can do a blind type of docking with Autodock and see, because loop regions and all could be flexible, so it might not be very stable also. So always alpha helix. First you do a blind docking and see how good the docking scores come out. Is there any drug reported for this particular target? Any drug reported?
No, sir.
Absolutely nothing.
No, sir.
Then how did you select this target? How do you know this is the target?
I choose based on the because it is essential for its survival. So if we target that protein means we can able to inhibit its growth.
Yeah. No. See, two questions. One is there any drug reported for that disease?
Yeah, general drugs reported for the disease
Is there any mechanism discussed for those
target.
not target? Is there any mechanism reported for those drugs?
Yeah, sir. Yes, sir.
So it is safer to look from that angle rather than going into this angle.
Okay.
If you don't have any drugs reported for this, and if you don't have any structures, nothing, then it's very dangerous. So you should start
Oh,
from the other way. So if there
yeah,
are
yeah.
any drugs for the disease, somebody
Mm.
might have done mechanistic studies for different those drugs. Then start from that angle. Okay, I
Okay, sir.
suggest start from that angle rather than this angle. Okay,
Okay,
because
sir.
here you don't
Okay.
know anything, so it's very dangerous. If you don't know anything, then it's very dangerous.
Okay, sir. Thank you, sir.
Yeah,
Then I have another one question, sir.
yeah, sure, go ahead.
Sir, I have heard a concept called reverse docking. That is most similar to what you talked in target prediction. That is Swiss model prediction.
Okay.
So,
Hmm.
what about do you know what about these things, sir? Can you
Oh,
elaborate, sir?
reverse docking. I don't know anything about reverse docking. See, the Swiss smart target prediction is useful for predicting side effects. Suppose if I have aspirin and it from the target prediction, it says it may go and bind to prostaglandin
Mm.
synthase 2 as well as it may go and bind to some other place. That means that could be a side effect, right? Because it is going and binding to some other target. So that
Yeah, 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 yeah.
you can try to predict whether there is going to be side effect because it is binding
Okay,
to some other target. So what we can do is we can modify the structure a little bit so that the binding with that another undesired target could be reduced. So from that angle, Swiss target prediction is very useful. Understand? So we
okay,
can modify a little bit of the structure and then again dock it, again see whether it is going to bind to both the targets or one
okay,
less
sir.
and one more. Like that, you can play around with it. Like a medicinal chemist, we can do a lot of playing around. So that is the usefulness of that. Okay.
Okay, sir. Thank you, sir. Thank you,
Thank
sir. Thank you, sir.
you.
So, good afternoon, sir.
Yeah. Go ahead.
Excuse me. Sir, I recently started to learn about docking. So my question is, like if we are docking any ligand onto protein,
Okay.
how we can be sure it would be inhibiting that protein or it would be activating that protein?
Okay. So, is there any drug reported for that particular protein? If it
Yes,
is
sir.
reported,
Yes, sir.
if there is first to do that, you dock the drug to the protein into the active site and see what is the energy it is giving. Okay, and it is reported experimentally, say it is inhibiting that protein. So the protein gets maybe down regulated or something. So that is your starting point. Understand? So when I design new molecules, something totally new, and then I dock to the same place, if the docking energy, binding energy comes out more negative, then you can say my molecules are better than the existing. So you should always start with the existing information and existing data. Okay.
Okay, sir.
Okay.
Yes, sir.
So
Thank
you start from that. So if you have a protein which you want to work towards for some disease, so somebody has already done created a drug, or a drug is there which is known to inhibit that protein. You start working on that and see how the docking takes place, what is the docking energy, where is the active site, what are the interactions. Then you, whatever designed molecules you have done in your uh, new molecules as a chemistry, then you, the chemistry person, you can start docking. So you already have a standard with which you work on. So always do that. So don't just blindly start doing anything. Okay.
you. Okay, sir. Thank you, sir.
Thank you so much, sir.
Okay.
Now we would like to enter into the valedictory session.
Okay.
Now we would request Dr. P. Haribabu, Associate Professor, Department of Biotechnology, to say a few words about it, the workshop.
Hello.
Hello, yeah, go ahead, please.
Hello. So this is Hari Babu, Dr. Hari Babu from St. Peter's College of Engineering and Technology.
Okay, good.
So we want to thank you, sir, first of all, for being able to give you a time for this kind of webinars. And we want to appreciate all the effort that you put into this one. And we want to thank all the participants that who spent 4 days in this time and then patiently waiting for each and every sessions and being responsive to all your teachings. We want to thank every single students who are helping us to organize these activities, and especially you, sir, yourself. We want to thank you for spending all this time for preparation, all those activities, and all those things. We want to thank you very much, sir. And hopefully, we can continue this process furthermore into the future if everything goes well.
What?
Thank you, sir.
Okay, thank you.
Hello, sir. This is Surya Lakshmi, Assistant Professor from Department of Biotechnology. Thank you very much, sir.
Okay,
Thank you for the valuable time. And also, I would like to thank all the participants, those who are actively involved in our workshop. Thank you so much, and thanks a lot. And also, I would like to thank everyone for the effort you made in this workshop, and also to maintaining the decent decorum in a public forum. Thank you so much. And based upon your attendance and participation, the certificates will be issued within 1 week of time to your registered mail ID. Thank you.
Thank you.
Thank you, sir. Tapna?
Yes, ma'am.
Let's wind up the session, ma.
Okay, ma'am. Thank you all the participants for your cooperation.
But 1 second, participants are requested to submit your attendance and feedback, which is displayed in the chat box.
Thank you all the participants for your great support to make this workshop a great success. Thank you all.